So I'm really happy to introduce Dr. Amy Lee and Dr. Olga Pacios-Santamaria to speak to innovations in vaccine development. Dr. Lee joined the Department of Molecular Biology and Biochemistry at Simon Fraser University as an assistant professor in 2020 and is also an investigator at BC Children's Research Institute. She's a Banting Discovery Awardee and a Michael Smith Health Research BC Scholar. Her group uses systems biology approaches to tackle global health challenges with the goal of improving neonatal sepsis diagnosis and combating antimicrobial resistance. Dr. Olga Pacios-Santamaria is a postdoctoral fellow in the Lee Lab with a background in antimicrobial peptides, their synthesis, and mechanisms of action. She currently works on the development of bacterial vaccines and LNP-mRNA drugs from an engineering perspective. Today, they're going to speak about accelerating the defense, rapid genomic surveillance of AMR pathogens with Oxford Nanopore to guide next generation LNP-mRNA vaccine developments. That's a lot of letters, but they'll explain them all, I'm sure. Welcome to the stage, Amy and Olga.
Okay, thank you, Natasha, for that fantastic intro. Olga, do you want to? Okay, you're going to take the podium. All right, we're going to work out the system. This is the first time doing a joint talk together. Okay, and then I have to figure out the slide thing. So I want to thank Katja and the bioMérieux team for the invitation to be here. As Natasha mentioned, a central theme of my research career has been using systems biology to sort of tackle diverse challenges, and I'll talk about some of that work, and I'll also explain what systems biology is. So we're very privileged to be on top of the Burnby Mountains, so that's where Simon Fraser University is on top of. It's on the unceded traditional Coast Salish lands, and you can see our campus is quite gorgeous. And as a fun undergrad project, I actually have my undergrad students go to the reflection pond and see what kind of Enterobacteriaceae they can find there and the antimicrobial resistance profile that you can get there. And then we sequence them. Maybe the bears play a role, but I don't know how that works. So in my lab, one of the central questions that we're really interested in sort of tackling is this antimicrobial resistance challenge. And of course, this is the audience where I don't really need to go into too much depth about how AMR is the hidden pandemic. We've heard the statistics over and over again that in 2019, 1.3 million people die from antibiotic-resistant bacterial infections. And that number is likely to be higher. There's probably 5 additional million people in 2019 die from resistant infections associated to resistant infections. And based on that estimate, what we're expecting is that more than 39 million deaths from AMR infections are expected and estimated between now and 2050. So that's an incredible number. Now, antimicrobial resistance impacts us all. And one area where it particularly impacts us is it really impedes how we can treat sepsis. And so I think we've heard discussion and mentioning of sepsis. I'm going to spend a little time talking about sort of the immunology of sepsis, where it's a life-threatening host response to severe infection. It's most commonly caused by bacterial infection, though viral infection can also lead to sepsis. And it really, again, sepsis is one of those diseases that can impact us all, but those who are elderly and who are very, very young are particularly at risk of becoming septic. And in my group, one of the questions and the challenge that we're focusing on is trying to understand why newborns are particularly at risk of developing sepsis. What we do know is that sepsis is very, very difficult to diagnose in infants. especially early on in the disease process. And unfortunately for newborns, there's really not a lot of time for you to figure this out. So clinically, they can progress very fast, and we're talking about hours to death. Right now, the gold standard for diagnosis is still bacterial culturing, right, which can take up to 48, 72 hours sometimes, which the babies just simply do not have. So we know that, the prompt treatment with antibiotics is probably essential for improving survival, but there's a lot of major consequences when you overtreat. And you can see this actually in global genomic surveillance work, trying to understand the resistance profile of bacterial isolates that cause neonatal sepsis. And this is a study called the Burden of Antibiotic Resistance in Neonates from Developing Societies. I'm going to refer to it as Barnard's, where they have 13 clinical sites, predominantly in sub-Saharan Africa and parts of Asia. And they enrolled 36,000 neonates who are suspected of sepsis. And out of the 36,000 neonates, only about 2,500 of them have culture-confirmed sepsis, right? So, what's going on with the other 80, 90% of those septic neonates who have no culture results, right? They're obviously very sick. And as a clinician, what do you do in that particular case? So what's interesting is this is focused predominantly on the gram-negative pathogens. So out of the 2,500 culture-confirmed sepsis, the dominant pathogen is actually Klebsiella pneumoniae, which will come up again and again. And it turns out that, if you look at all these pathogens that cause neonatal sepsis, they're resistant to the first line of empiric antibiotic treatment that's recommended by WHO. So the first line is ampicillin, and that's over 95% resistant. So whatever we're giving to the babies aren't even working. So where does my lab come in? Well, we take this sort of holistic approach called systems biology, where we really look at the biological challenge from a diverse perspective. So we think about it from trying to understand how the newborn immune development and immune response to infection can lead to sepsis. We look at it from the pathogen, so particularly in my lab, we focus a lot on Klebsiella pneumoniae. And we try to understand what are some pathogen factors that drive severe outcome, and how do resistance genes transfer. And we think about sort of the microbial composition, the microbiome, and how do the pathogen kind of function within this microbiome. And really, how systems biology work is that the entire system is much greater than sum of all of its components. And we use advanced omics technology, machine learning, different bioinformatics strategy to really try to develop better diagnostics, better therapeutics, and also the topic of today's talk, potential preventative strategies. So I'm going to take a little bit of time actually talking about how we apply systems biology in thinking about diagnostics. So in this case, we have a Malawi cohort, and similar to what we've seen in the Barnard study, our Malawi cohort have about 400 to 450 newborns, and they're all clinically diagnosed as having sepsis. But only about 20% of these babies have culture result. So again, 80% of these septic babies are culture negative. So what do you do as a clinician, right? You err on the side of, here's a very sick baby. You have to give the baby something, right? And so that's where antibiotics are typically given. But what if that baby has viral infection, or is just not feeding well, or has jaundice? And so one of the ways that we're kind of dealing with this is we're using machine learning from the host perspective to see if we could find host biomarkers, immune biomarkers, that can identify those neonates who are true septic. And then another way that we're doing this is from the same samples, we're performing sequencing to see if we can detect bacterial genes in the blood. For those of you who might be interested in hearing more about that pathogen diagnostic aspect and some of the assays development that's been done, go check out Claudia's poster number 44. She's in Dr. Lavaugh's lab, and Dr. Lavaux is a neonatologist, and together with Dr. Lavaux, we've been really working on sort of leveraging our different expertise to try to develop both a host immune as well as a pathogen biomarker that, can sort of give us a more comprehensive diagnostic rather than just relying on one or the other. So where are we going with this? We know diagnostics is a very critical aspect of preventing indiscriminate use of antibiotics, but we also understand clinicians are under immense pressure to sort of figure out what's going on with the newborns. So our goal right now is to sort of apply advanced machine learning, integrate molecular data and clinical data to yield sort of meaningful diagnostic biomarkers. We're working with NRC, Dr. Veres at the National Research Lab to develop some lab-on-the-chip prototypes for host biomarkers, not just for newborn, but for adults. And we think that this better prioritization and more targeted antibiotic treatments will allow us to develop, better prioritization and diagnostics will lead to more targeted antibiotic treatments. But at the end of the day, right, prevention is better than cure. Again, I don't need to belabor the point, but we all know WHO has come up with a bacterial priority pathogen list for those highly resistant pathogens that are in urgent need of R&D, particularly in the drug development space. And this is not surprising, 6, and actually really if you count them all, 12 of the leading AMR pathogens are on that priority list. But when we think about R&D from a drug perspective, it ultimately will lead to resistance. And so we're kind of in this circular loop, right? So, you might ask, okay, well, how about vaccines? So I was reading the report from Vaccines for AMR.org, which is a collaborative work between Boston Consulting Group and Wellcome Trust. And you can actually see that for something like Klebsiella pneumoniae, where the urgency of AMR threat is 2 being the highest score, the pipeline robustness for vaccine development and our understanding of pathogen biology, as well as some of the preclinical success rate, is near 0. Okay, and it isn't just for gram-negative pathogens, it's also true for gram-positive pathogens. So a group of us in 2023 came together and said, well, can we develop a robust vaccine pipeline against bacterial pathogens? And this is where Avenger comes in. Maybe now the cartoon in the beginning makes sense. I'm not going to read out our abbreviation, but you can see we labored over the name of the project so that we could fit Avenger into it. It's a large consortia that is co-led by Dr. Blakeney, who is an expert in self-amplifying mRNA, and Dr. Peter Colas, who's an expert in the LNP technology. And there's a diverse team with expertise across systems immunology, vaccinology, bacteriology, as well as in thinking about elderly immune responses, immunosenescence, how do we develop vaccines that can really target different population, as well as the key critical part is that this vaccine project is going to develop in parallel a biomanufacturing capacity. So we'll have, at the end of 4 years, not just vaccine candidates, but actually a pipeline to generate potential vaccines against AMR pathogens. So the goal is, hopefully we can build a robust pipeline that allow us to have 100-day response. And the first aim is really to develop a high-performing, fit-for-purpose RNA LNP vaccines that have improved efficacy for at-risk populations. And we'll define that as we go along. The second aim is we're going to find the vaccine targets and candidates against 3 critical AMR priority pathogens, and they're listed here: K. pneumoniae, strep pneumo, and Staph aureus. And then the last aim is to have a biomanufacturing pipeline set up, and we'll test it in a non-human primate infection model. So what are some of the challenges involved in making bacterial vaccines using the LNP-mRNA platform? I don't have all day, so I'm not going to list them all. But some of the key challenges that we're thinking a lot about is how do we select the best bacterial antigen? And this antigen might be different depending on whether or not you're an intracellular pathogen or extracellular pathogen, or you are at different tissue sites. How do you express bacterial antigens in the mammalian system, in the human cells? And what constitutes as a protective immune response? Are we gearing more towards humoral responses versus the cellular responses? And what is the appropriate preclinical animal model to assess our efficacy of the vaccine? So I'm going to hand off the rest of the talk to Olga, who actually does the work in the lab, and she will walk us through some of our solutions for these challenges.
Thank you very much, Amy. So yes, just to dive a little bit into the first challenge that Amy just mentioned. So first challenge, okay.
Do you want me to do it?
I have it now. Yeah.
Okay, cool.
Perfect.
One more.
Thank you. Yeah. So first challenge would be the huge bacterial diversity, right? So just to illustrate this, for instance, if we take the tree of life, where we have the representation of all the living beings, you can see that half of the tree of life is actually represented by bacteria. So the diversity is huge if you compare to this tiny little green symbol there, and the thickness of that symbol corresponds to not some animals, not some fungi, but all the animals and all the fungi. So really, this comparison is very illustrative of the huge diversity that we are facing when we deal with bacteria, especially clinical bacteria. So this brings me to the idea that if we take 2 strains of bacterial isolates and we compare the genome similarity, we can have roughly between 60 or 70% genome similarity. Whereas if we take 2 humans, the similarity goes up to 99%. And so if we zoom in the bacterial part of the tree of life, and we take, for instance, Klebsiella pneumoniae as an example, you see that the genome, diversity in the phylogeny here represented is really huge. So this is something that we need to bear in mind. And actually, a very important realization that Jonathan Ho, bioinformatician at the Lee Lab, did was that if he took the RevSeq genomes deposited in the NCBI database, the gene bank database, and if he was only working with these genomes, he saw that they are not at all evenly represented on the database. So for instance, you have the vast majority of the genomes belonging to the ST11, whereas other very clinically relevant genomes are not represented in this data set. And very importantly for some pathogens as Klebpneumo is the fact that some isolates contain a large number of plasmids. For instance, in this plot here, you can see that some strains can harbor till 10 plasmids, and the vast majority of the genomes, again, have 3 plasmids in each cell. So this is it's really important for us, and this illustrates that we really need to sequence what's going on out there and what's causing infections nowadays. So we need to have this contemporary sense of what's going on, and we can't solely rely on what's deposited in the public databases if we want to make a protective vaccine. So to overcome this issue, what we're using at the Lee Lab is this long-read sequencing platform called Nanopore. And so it's really different compared to the Illumina sequencing, which is based on short read. And what Nanopore does is it actually represents a very reliable technology to, for instance, assemble large plasmids, as for the case of Klebsiella pneumoniae, as I just mentioned. And the technology is, if we take, for instance, this flow cell here for the MinION instrument, it contains an array of pores that are embedded into a membrane, and this membrane separates 2 ionic solutions. And so the way this works is that this pore here, whenever the DNA strand passes through the pore, the nucleotides are going to disturb the voltage that is applied at both sides of the membrane. So in these 2 ionic solutions, there is this difference of voltage, and the voltage is going to be proportional to or associated to each nucleotide that is passing through the pore. So this is recorded by the instrument, and so the perturbation or the changes in the voltage are associated to a particular nucleotide, and this is how we can get the sequence of nucleotides that we are seeing. And this technology is real-time and at 400 nucleotides per second. And actually, we're not using MinION anymore. We're using PromethION, a P24 instrument, which is very big, very powerful. It handles 24 flow cells at a time. In each flow cell, we can sequence 96 bacterial genomes. So in one sequencing run, we can get something around 2,500 complete bacterial genomes with a very good depth and very good coverage of sequencing. So pretty reliable and robust technology. So our progress so far, in the lab, we've received something around 574 clinical strains, and these all come from sterile sites or the blood, so these are invasive isolates. So far, we've gotten Klebsiella pneumoniae and Staph aureus, and we have streaked out all of them, pelleted, froze them, extracted some of them. We do extractions at 96 well plates, so it's a 96 well format, so it's pretty high throughput, I would say. Then we do quality control of this DNA. We need to ensure that the DNA is, there's integrity, there's high purity, because Nanopore is really extremely sensitive to the purity of the DNA that we use as an input. And so after all these quality control steps, we sequence them. And we've sequenced so far 150 isolates, so we're getting there. So these 4 graphs provided by Jonathan, They just illustrate the quality of the sequencing run. I took here one of the last sequencing runs that we did at the lab as an example. So 50-ish clinical strains of Klebsiella pneumoniae were sequenced in a Promethean flow cell, as I showed you on the last slide. And so here you can see the total bases sequenced. The N50, which is a parameter that measures the quality of the run, so the larger, the better, and it's actually the length of the largest contig that together with the contigs that are equal or smaller than that contig contains the 50% of the total genome assembled. Then there are these histograms here representing the length of the reads that we're sequencing. We're doing a long read again. And then the coverage. So we aim at a 30x coverage for each strain. So this is the dotted line. And you can see that most of our isolates reach this threshold. Okay, so what do we do with all this sequencing, right? What do we do with the sequences that we get from invasive isolates and from clinical strains that we recruited in the hospital? So it's important that we do all this, but what's the point in the vaccine, right? So this is not the subway map. It's actually the reverse vaccinology pipeline that Travis and Jonathan, the two bioinformaticians at the Lee Lab, developed in order to get a handful of candidates that will be used as antigens for the vaccine. So the way this works is they were using primarily NCBI isolates, but the idea is that they will incorporate all the sequencing that we're doing at the wet lab. And they're doing annotation of the bacterial genomes. They're doing a pangenome analysis to get the core genome, so all the genes that are present across all the clinical strains that we're considering. And they're running these 3 steps sort of in parallel. So the identification of extracellular targets and surfacely exposed antigens, because we want, of course, that the host immune system will recognize the pathogen whenever we get infected. Then they're ruling out the candidates that will share homology with human proteins, and they're ranking the candidates based on entropy scored and the divergency of the sequences across the conserved genes on the dataset. Then they will try to gather some expression studies on transcriptomics and proteomics, ideally in vivo, and some strain metadata when it's available, and then they will pass along this list to structural biologists, such as the Strinatka Lab and others at UBC, that will make some 3-dimensional predictions and some structural studies in order to ensure that the antigens are folding and they're functional. So one can, at this point, wonder, why don't we have as many bacterial vaccines as we do have for against viruses, right? And this might seem obvious, but actually there's a huge difference in terms of complexity when we compare viral pathogens and bacterial pathogens, both at the structural and the genomic level. And so if we think about SARS-CoV-2, for instance, as an example, we only have one or a handful targetable antigens on the surface, such as the spike protein. However, if we take a gram-negative, such as Kleb pneumo, or a gram-positive, such as Strep pneumo or Staph aureus, and you see the cell wall, the complexity of these surfacely exposed antigens is huge. And we have lots of antigens that might be good candidates, but also we have polymers that might hinder the B cell and T cell epitope recognition, such as the capsular polysaccharides, and that might mask some antigens. So this is not at all a trivial part of it. And then the genome size is not really comparable, right? SARS-CoV-2 had like 29 kb, and 29 kb for Kleb pneumo is only the operand that encodes the capsular polysaccharides. So, So it's the whole genome of Klebsiella pneumoniae is almost 6 megabases. So yeah. And then very important, as Amy was saying, is the fact that viruses rely on the host cell's machinery in order to express and translate their genome. However, bacteria have their own machinery. And whereas in mammalian cells, glycosylation and other post-translational modifications are very common, it's not the case for bacterial proteins. So more challenges. Okay, so let's assume that we overcome these issues and we have a short list of good candidates and we have selected the right antigens. Now, how do we make sure that if we engineer these antigens in an mRNA and we encapsulate that into an LNP, how are we gonna elicit immune responses that are actually protective? Well, there's lots of research going on and lots of optimization going on, especially in the Colliers lab at UBC and others. So just to summarize here, 3 different types of LNPs, but this is actually huge. One of these is these kind of bleb-like structures. These are ideal for a vaccine perspective. So they have this hydrophobic core in the inside and this hydrophilic compartment where the mRNA is going to be encapsulated. And then we can have also some long circulating systems playing with these ionizable lipids and helper lipids so that they will extend the circulation half-life in the body systems. And also we can have this kind of Onpattro-like structures, which are great for encapsulation of siRNA or small interferon RNA if we want to knock out a gene, but they're not really interesting in the context of mRNA vaccines. So Muzammil Khan, a postdoctoral researcher at the Colliers and Russell labs, he came up with a formulation called here the LNP. It's fuzzy. The resolution in the original slide is a little bit better. The LNP here. And so he's measuring luminescence because he's using as a cargo an mRNA that is encoding the FLAC or a luciferase. And so he's measuring the transfection capacity and the expression of the cargo that he's using. And you can see here that this LNP formulation, compared to others, such as MC3, this is the ionizable lipid that he's using, or ALC in gray, and this is the ionizable lipid included in the Pfizer-BioNTech vaccine, target specifically lymph nodes and spleen. And this is very important in the context of a vaccine because for sure we have the professional antigen-presenting cells and immune cells that might be relevant for our outcome. And then to make things even harder, we also can have lots of optimization and lots of research going on for the RNA. So especially the Blackney and the Jens lab at UBC, they're doing some research in the cargo that we can use the cargo that we can use in the LNP-RNA vaccine, right? So we can have the mRNA vaccine, like a conventional molecule, but the idea here is to do the multivalent approach because for sure we're targeting huge genomes, as I mentioned before. So if we do a multivalent approach, we will have different antigens encoded in the mRNA. And the idea is to encapsulate them in an LNP, and then they will get translated into the mammalian cells through the ribosomes, and the half-life of the protein expression is actually limited to 3 days. And another limiting factor is that what we can encapsulate as an mRNA is only 2 kb. However, for self-amplifying RNA, we can lower the dose because other than the antigen, they will also express these RNA polymerase that will self-amplify the molecule. And for the circular RNA, this is a very promising field, but it's still very experimental. And the idea here is to overcome all the stability issues that are associated with the RNA technology. Okay, so this slide just summarizes everything we've just said. So we're focusing on these 3 pathogens. We are recruiting clinical isolates. We've started the sequencing. The reverse vaccinology pipeline is up and running. And once we have the best candidates, we will clone them. We will express them as RNA. And we'll try to have the best RNA and the best LNP for the vaccine purposes. And then once we have that, we need to have a suitable animal model in a preclinical stage to test this. And then the next steps will be to characterize this response in some other populations, such as the aged and the frail, and to keep on building these pipelines, to keep on working on the RNA engineering and the LNP side of it. And yeah, lots of work to do, but pretty exciting. Hopefully we have some candidates soon to start to play around with them. And then all of this work that Professor Lee and I presented would have never been possible without the help of the funding agencies and, of course, all of the people who have been involved and done all the work. So thank you very much for your attention.
Thanks so much, Amy and Olga. Any questions in the room?
Thanks, very exciting, and we're sharing our clinical strains with the project.
Yeah, 100%.
Interesting to think about a lot of neonatal sepsis in developing countries as culture negative. In fact, many of them may not have access to culture. So if they were in a rich country, they probably would have a positive culture.
Yeah, that's you're totally right, and that's part of the challenge in sort of how we're developing the rapid diagnostics. Now, the two studies that I presented, the Barnards that had about 35,000 newborns, and the Malawi cohort with Pascal Lavoie, that's roughly about 450 newborns. Those two studies, we actually brought in culture bottles to do the experiment. But you're so right. We have clinical sites where we cannot get culture results because they don't have the machine to incubate the culture bottle. So, you know, this increases the challenge of diagnostics, right? And I think, you know, earlier you and I were chatting about the importance of how perhaps molecular diagnostics can sort of deal with the equity challenge here, but the cost needs to be less than the cost of the treatment. So this is, at the end of the day, some of the struggle that we're dealing with, for sure.
Hello, thank you. Very interesting project and ambitious.
Yeah.
So, just a question. You know, you have mentioned that you are sequencing all the clinical isolates. You are aligning them, doing phylogeny, and finding the common epitopes for generic vaccines. And so in that way, you're shortlisting your candidate antigens. But how are you actually further shortlisting them based on like the immunogenicity or all these things? So this is one thing. And second thing is, I noticed in one of your slides you mentioned you're also doing the host transcriptomic analysis on the same patients. So are you planning to get some like information on the immunogenicity or epitope prediction based on the transcriptomic data?
Yeah, so I'm going to answer the second question first. So in the septic neonates from the Malawi cohort, we only have 8 newborns who have confirmed Klebsiella pneumoniae infection, right? So that's not enough to bank anything on. But you know, you bring up a good point, right? In how can we use the host immune responses to kind of inform vaccine design, and this is something that we're definitely looking into. We're working with, as you saw in the Avengers slide, we have 14 labs across Canada. And so we're working with structural biologists, but we're also working with experts in B cells and T cells. And so we're doing a large number of studies to try to understand, you know, that what makes the best antigen.
Hi. Fellow engineer here who has a specific interest in Klebsiella pneumoniae and using machine learning to try to do a lot of the same kind of stuff that you guys are doing. So also that was a good question to lead into
Sure.
what I want to ask. Are you using any sort of machine learning in the when you're trying to shortlist some sort of antigens?
Yeah, that's a really good question. So we use machine learning in our host work because in the host transcriptomic, you know, again, we're using supervised machine learning. We also have unsupervised machine learning, but in the supervised world, you need good labels, right? And so we can use, say, culture result as a label. What would you use as a label in Klebsiella pneumoniae? Let's give it as an example. We know it's a colonizer, but sometimes it can be invasive. But what makes them invasive? And what makes them stay as a colonizer? There's lots of really interesting questions around there, lots of interesting biology around there, but there's not a good label, right? Because again, you have to think about, is there kind of interaction with the host, right? So there's a large study done, for example, in the UK, where they show that ICU patients who succumb to Klebsiella pneumoniae sepsis, they can actually find Klebsiella pneumoniae at hospitalization in their gut microbiome. Okay, so that idea of colonizer versus invasiveness, is it context dependent? So it makes machine learning very tricky, and that's why our vaccine design is a very rationalized approach, really thinking about, can we find targets that conserve across as many Klebsiella as possible, ideally found predominantly in the invasive isolates that we have in the lab, but we're not going to exclude them necessarily because of colonizer. But we also have to think about, what does that mean in terms of the microbiome and so on, right? So it's, I don't know, it's kind of a rambling non-answer, but maybe we should chat after with over beers.
I like the sound of that.
Okay.
Thank you.
One more round of applause for Amy and Olga. Thank you. Thank you very much.